This video is split into 4 sections to help you understand base editing using CRISPR-Cas9. I will talk about nCas9, the 2 primary types of base editing, their applications, and big limitations. Generally, a Cas9 is functionally split into 2 lobes, one for recognition and the other for cutting up DNA, but the linear polypeptide of a Cas9 looks something like this. The RUVC nuclease domain is not one continuous string of amino acids. The HNH and PAM interacting domains are, however, simpler. From my previous video, you may recall that disrupting the 10th and 840th position results in a dCas9. Instead of creating 2 mutations, what if you only mutate the RUVC domain? Alternatively, you can have a Cas9 version with a mutation in just the HNH domain. These are what we call nCas9. The first version, the one containing mutation in the RUVC, can only cut the paired DNA strand using the active HNH domain. The other version, which inactivates HNH, can only cut DNA using the RUVC domain. So instead of getting a double-stranded DNA break, you end up with a single-strand cut, called a nick. And this is what the N stands for. The D10A or RUVC mutant is quite commonly used in base editing. The other one, the HNH mutant, is fine too, but 840 alone does not kill the activity of the domain. You need a second mutation for 100% inactivity, so it is a little more nuanced. Base editing relies on DNA repair. You will see that in a moment, and for that reason, nCas9 is always preferred over dCas9. Base editing is simply a strategy aimed at changing nucleobases at a single base resolution. Think point mutations and a way to fix these point mutations. Of the 2 primary types of base editing, the first one involves changing adenosine into an inosine through deamination. The inosine is then read as a guanosine by polymerases. The enzyme responsible for removing the amine group is an adenosine deaminase, and this base edit converts an A to a G. The second base editing type acts on cytidine and attempts to convert it into a uridine, which is essentially thymidine. Again, a cytidine-specific deaminase is involved in this reaction as well. Let's go into some details and start with cytosine base editing. The idea is quite simple. You have a nCas9 targeted to a location through a guide RNA. This location contains the cytosine on the non-pairing DNA strand within the R-loop structure. The deaminase enzyme is fused to the nickase at the N-terminal end, which deaminates the cytosine on the unpaired strand. And the cytosine is converted into a uracil base. Because a nick is made in the pairing strand, a short-range resection starts, and DNA damage repair proteins gather around and resynthesize the nicked strand. After repair, the original G is replaced with A using the uracil as a template. Finally, the uracil is converted back to thymine during replication or through a base repair mechanism. This is the ideal C to T base editing pathway. Unfortunately, there are some repair proteins in the nucleus called uracil glycosylases, and their job is to remove uracil from the DNA, because uracil does not belong in DNA. So naturally, the newly formed uracil is removed and cytosine is swapped back before the base edit can resolve itself. Sometimes it is also possible that excision repair just removes the GU base pair altogether, which can result in a deletion. A way to counteract this cellular repair pathway at the base editing location is to fuse a protein called uracil glycosylase inhibitor to the C-terminal end of nCas9. This ensures that excision and repair proteins do not interfere with the edited base, and the probability of your desired edit becoming permanent increases drastically. The cytidine deaminase, by the way, is called ApoBec1. As you can see in the name, it is an RNA-specific enzyme. Fortunately, it also has a single-strand DNA-specific activity. And in this 3-way R-loop structure, you have this unpaired single-strand DNA. So this RNA-specific enzyme just works without any issues. Although most current versions of this enzyme are engineered for efficiency and DNA specificity, the principles of editing, however, remain just the same. But here's the actual problem in base editing. In an optimal guide targeting region, you expect a 50% GC content. Then this begs a question: which cytosine in the DNA is getting the edit? Or rather, if I want to specify the cytosine for edit, what needs to be done? It turns out that the base editing happens in a small window in this R-loop structure that spans 20 nucleotides. In the first iteration of base editing, the default editing window for cytosine base editors was between position 4 and position 8 from the 5' end of the guide region. If you do the math, it gives you about a 5 base window. Not super specific if you had expected a single base resolution. More recently, the editing window has come down to 2 or 3 bases, but it is still not at a single nucleotide level. The editing window is largely a function of the linker length, the Cas9 modifications, and the deaminase engineering. So the key takeaway here is that cytosines close to the PAM or 3' end of the guide RNA are not edited, and the extreme 5' end is also not edited by the enzyme, and the deamination occurs on the non-nicked DNA strand. Let's contrast cytosine to the adenosine base editors. The concept is fairly similar to what you just learned, except that the adenine is converted into inosine, which is eventually reverted to a G after the repairs are complete. In this case, again, the N-terminal end of Cas9 is fused to the deaminase enzyme to achieve the base edit. But there's a problem, or rather, it was a problem. You see, there are no naturally existing adenosine deaminases that function on DNA as a substrate. There are plenty of RNA-specific adenosine deaminases, and one of these enzymes called TAD-A came in handy. It was taken from E. coli, and that is the enzyme used in adenosine base editing. But before we can use this, researchers evolved and engineered the RNA-specific enzyme to lose its RNA specificity and gain DNA specificity, and that engineered version is what gets used today. There are many alternative RNA-specific enzymes that people have evolved and engineered, but TAD-A is most widely used. And if we ask the same question about editing windows for adenosine editors, the same logic will hold true. In the first few iterations, the editing window for ABEs was between 3 to 4 bases, but in more recent versions, it is quite close to a single base resolution. A big change in adenosine editor development comes in the form of a dimerization of TAD-A. Current versions contain 2 monomers of TAD-A in tandem. The wild-type non-engineered TAD-A is present at the external side, and the engineered TAD-A is present on the internal side of the N-terminus. The reason for creating this dimer is because TAD-A works in a dimer, and structurally it is much more efficient for base editing to artificially force dimerization by having 2 monomers in default proximity. Now, when we think about application of base editors, the obvious one is to induce single base changes that can fix diseases. Outside of that, let's think about CBEs and these set of codons. These codons can be converted into stop codons to result in a premature termination of a protein product. This application is called CRISPR stop. Alternatively, if a stop codon is present in a diseased condition that may result in a toxic protein, the ABEs can convert stop codons into a coding segment to hopefully reduce its effect on the disease. This type of application is called crispr-Cas. ABEs can also be used to convert start codons into non-start codons, and that way a protein is never expressed. This type of strategy is called I-silence. Just like this, there are many other applications for splicing control, drug screening, and mutagenesis where base editing can be useful. All this sounds great, but there are some serious limitations to the current standards of base editing. Typically, epigenetic modification of bases, like methylation at CpG islands and other similar chemical modification, can reduce the efficiency of base editing. For CBEs, we know that there are some specific sequence biases in ApoBec1. Cytosines that are present next to a thymine are more likely to get edited than others. And hopefully you have noticed the concern regarding unintentional base editing given the multi-base long editing window. For instance, if you have the CCT in a base editing window and you want to edit the first C, then it is possible that the adjacent C also gets edited. This will be an undesired bystander edit. it is possible that the first A gets edited alongside your target adenine, if they are in the same editing window. So given some context, single base resolution of base editing can be difficult to achieve in some situations. Also, given current methods, there are only two primary editing methods, purine to purine and pyrimidines to pyrimidines. Therefore, only two complement pairs can be successfully edited. There is a C to G base editing system, so in principle you can do that as well, but they have their own nuanced issues. But even with all this, only the transitions and maybe one transversion is all we can do for now. Let's take the example of sickle cell anemia to understand this better. Compared to a normal case, the diseased state often contains a point mutation in the hemoglobin gene that results in a specific valine codon. This mutation is a purine to pyrimidine switch. As we just noted, the base editors cannot convert between purines and pyrimidines. You can use something known as dual base editing system, but even that wouldn't work in this case. Instead, you can potentially install a new base edit and mutate somewhere else in the hemoglobin gene or its regulatory regions in such a way that a new mutation counteracts the original mutation. This would be either a compensatory or perhaps a suppressor mutation, where the hope is that two negatives come together to rescue the original defect. It fixes the diseased state, but it does not necessarily fix the original mutation in the DNA. Unlike sickle cell anemia cases, maybe in another disease, a compensatory or a suppressor mutation is not possible. In that case, maybe a transversion is absolutely required, and maybe targeting and fixing the exact mutated base in the DNA is the only solution because any other approach would not work. What do you do then? In those cases, perhaps prime editing is the answer, and I will discuss that in the next video.